All right, I'd like to introduce our next speaker, and it is my great pleasure and privilege to introduce Dr. Michael Friedlander, who is the founding executive director of Virginia Tech's Carilion Research Institute. He's the associate provost for health sciences and the senior dean of research at the Virginia Tech Carilion School of Medicine. He provides leadership to more than 24 premier research teams, developing new strategies for diagnosing and treating major health challenges. As Virginia Tech's Associate Provost for Health Sciences, he leads the development of the new Faculty of Health Sciences and the university-wide doctoral program in translational biology, medicine, and health. He's also a professor of biological sciences, biomedical engineering, senior dean for research at the School of Medicine, professor of psychiatry, behavioral medicine. Very impressive. Before joining Virginia Tech, he was at Baylor. We thank heavens we've gotten him here in Virginia. He actually has a very unique connection with this show and this event. Turns out that he is David Eagleman is the scientific grandson of his. So he trained Reed Montague, and Reed Montague trained David Eagleman. He has recently returned from the Antarctic, where he has been working on Arctic fish with his techniques of looking at plasticity in the brain. He is going to talk to us about new levels of understanding of brain function for better science-based diagnoses and treatment in Virginia, the brain state. Thank you.
Thank you very much. It's a pleasure to be here. As was mentioned, David Eagleman, who's running the show that I think a lot of you have been watching, trained as the first student of my former first student, and so I'm still very connected with David, and I hope many of you are watching the show. I think it's been absolutely fascinating. What I'm going to try to do in my limited amount of time is introduce you to this concept of our state, Virginia, as the brain state, talk a little bit about the exciting brain research that's going on at the new institute we founded, and then I'm going to focus in at the end on a little bit of my own research on trying to rebalance circuits similar to what you just heard with a little bit of a different twist. So let me just start out with this quotation made by Max Cowan, the founding chief scientific officer of the Howard Hughes Medical Institute, and Eric Kandel, Nobel laureate, who said, "Disorders of the nervous system taken together basically account for more cost and care than all other disorders combined." True statement. You can lump all together all cancers, heart disease. They are eclipsed when it comes to disorders of the nervous system. And so a little bit of the data backing up that statement are shown here. There are well over 600 disorders of the nervous system. Costs the United States economy about $1.4 to $1.7 trillion per year. Here are some of the facts and figures on just a few of those. I'm not going to go through these. I think many of you are familiar with the burden our country has with respect to addiction and substance abuse, autism, Alzheimer's disease, with their tremendous increase expected with population aging, deadly forms of brain cancer, mental illness affecting 60 million Americans, 13.5 million of those serious mental illnesses. You just heard about Parkinson's, other neurodegenerative diseases, and of course traumatic brain injury. So this is big stuff for the United States and the world. It's an important mission for individuals, families, and society to address these issues, and I think we're all obligated to be involved. You've heard a little bit about this mention in Virginia, the brain state. I'll simply say that a number of our institutions are working together with state government and private industry to try to tie in our resources into an effective collaboration unparalleled in the United States in terms of brain research. And we're addressing many areas. A few of those are listed here: addiction, brain injury, intellectual disabilities, nervous system cancers, neurodegenerative disease, psychiatric disorders, etc. By bringing together the strength of an outstanding cadre of investigators from all the institutions of this state, we can do something that really is unprecedented. And I can talk more about that later for those of you who may be interested. So I want to tell you now a little bit about what we tried to create in Southwest Virginia with the new brain research, series of brain research programs. I'm going to highlight some of the work of the other investigators there first, particularly initially starting with addiction, led by Dr. Warren Bickel. Dr. Bickel studies temporal discounting, or how our brain values things into the future. If you take anybody in this room and evaluate how long things last in terms of having value, Dr. Bickel does a pretty simple test. On average, you get 4.5 to 5 years to value approaches 0. If you do the same test with a heroin addict, 7 days. After 7 days, nothing has any value. You can offer a heroin addict $1,000 to come back in a month to the lab and get it, real money, or $5 to take now. They'll take the $5 every time. And it's not just heroin addict, it's all kinds of addiction. Our ability to assign value in the brain is somehow corrupted. And Dr. Bickel and his colleagues have found ways to actually therapeutically intervene to change that valuation function, affecting not only addiction and substance abuse, but other health behaviors such as overeating and obesity, for example. In addition to this sort of behavioral work, we have a number of investigators that are using functional brain imaging. You heard about fMRI to try to help people to control addiction. So this is an image of a human brain, front and back. The highlighted areas show areas of statistically significant change in blood flow, we think as a proxy for the neural activity in that area based on oxygen delivery. And those are being activated as a subject in the scanner is craving, in this case, cigarettes. This is a smoker who's been shown pictures of cigarettes while they're in the scanner. This circuit in the brain lights up, becomes highly active. They start to sweat, lick their lips, and get fidgety, and they need a cigarette. This is the craving signal in the brain. You can use the brain imaging signal in a feedback mode to actually teach the person to tamp down and decrease the craving feeling in their own brain. You can retrain the brain to manage that, as you can also retrain using real-time fMRI the brain after stroke or traumatic brain injury to recover function in otherwise quiet areas. And so this is some of the work that Dr. LaConte, who does this imaging, has done. I'm going to show you a quick little movie clip. I hope. You know how these things go. Okay, what you're looking at, I know it's hard to see. You're looking down on a person's brain. You may see some twinkling blue and red spots. These are areas of the brain becoming active as the person is thinking about trying to move this cursor on the screen while they're lying in the MRI. This is tracking the movements. What Dr. LaConte has done is analyzed all 30,000 little voxels in the brain simultaneously to generate a vector that the person is thinking about where they want to move the device. And he's coupled the output of the person's brain to the machine to drive the device. He's also used this to drive cars. We have people in scanners in our building who are thinking about driving a robotic car. It's on the ground floor of the building that rides around. They can navigate it through the streets. So you can think about ways to interface the human mind with devices in a variety of ways using this technology. I also mentioned that we have a considerable amount of work going on with brain cancer. I want to highlight the work of Dr. Harold Sontheimer in that regard. This is in a very aggressive form of malignant brain tumor known as a glioblastoma multiforme, or GBM. Basically, the outcome is virtually always bad. This is something called a Kaplan-Meier curve that shows you the survival from time of diagnosis and how many people still remain surviving. And as you can see, it diminishes quite readily. Regardless of what resources you have or how good your neurosurgeon is, the problem is the malignant brain tumor cells that are a type of cell called a glial cell will shrink down and migrate through very small spaces in your brain. The space in the brain is very tightly packed. And what Dr. Sontheimer and his colleagues found is these glial cells will express a certain sort of a pore or a channel when they become malignant that allows salts and water to move across the membrane that lets the cell flatten out like a sheet of paper, actually much thinner, and it will migrate to other spots in the brain. So no matter how good the neurosurgeon is at this point, the brain has already been seeded. What Dr. Sontheimer and his colleagues then discovered was the giant Israeli scorpion has a venom in its tail that has a molecule he named chlorotoxin because it interferes with the channel, which is a chloride, as in sodium chloride channel, on the surface of the cell that allows the shrinkage to occur. He then synthesized and purified this very complicated molecule through a company he set up, and then he took it into clinical trials in humans. These are pictures from the first trials showing it bound only in the brain and only to the site of the tumor. This is the urinary bladder, and what they found is it extended life fourfold in the patients that went into the second phase of clinical trials. It's not a cure, it doesn't make the brain tumor go away, but it stops dead in its tracks the migrating cells from seeding new areas until some other therapy might be applied. So it's a step in the right direction. I mentioned functional imaging, but I want to tell you a little bit about something we do with it that's a bit different. These scanners we have are interlinked together throughout a network we have in Roanoke, Virginia, and all over the world and the country. Our scanners are linked in up and down the east and west coast of the United States, Princeton University, Caltech, Baylor College of Medicine, Emory, a number of other places. In Asia, at Hong Kong University of Science and Technology, Yonsei University in South Korea. In Europe, in London, in Ulm, Germany, in Oslo, Norway, etc. And this interactive network developed by Dr. Reed Montague allows for functional interactive brain imaging to diagnose and be able to develop newer treatments for psychiatric disease that are heretofore unrealized. Because you can see what one brain is doing and another brain simultaneously as they interact in a social exchange. And this is one of the things that breaks in a whole bunch of psychiatric diseases. So Dr. Montague and his colleagues have used this in a variety of ways. The most recent is illustrated here, where they published this work a few months ago, where they developed a 4-minute fMRI test for autism in children. Non-invasive, no ionizing radiation. The child simply lies in the scanner, looks at a series of face pictures of themselves, family members, others, and by virtue of mapping the activity along an area of the brain right here known as the cingulate cortex, they discovered there's a map of what's called self and other in that region that is inverted in people when they have autism. World's first quantitative, precise, mathematical diagnostic for autism, not relying on a survey, a questionnaire, or a patient report, but relying on a real quantitative measure. In addition, you just heard about deep brain stimulation, a fantastic new therapy, as you saw. Our investigators, led by Drs. Kashida and Montague, have developed a way to piggyback with the stimulating electrodes you just heard about and recording electrodes, a microfiber made of carbon that can be lowered down by the neurosurgeon and measure in real time, for the first time ever in a living human being, the release of the chemical dopamine you heard about, which provides a reward signal to the brain and is so important in behaviors in addition to the motor behaviors you heard about as well. This can now be measured, and real feedback can be provided to the clinician about what's going on in terms of modulating that activity in the brain. Now, I'm going to end up by telling you a little bit about my own research in my laboratory that relates to a few of the topics I've alluded to and you've already heard about. We are particularly interested in traumatic brain injury and how it might be possible to rebalance, like you heard earlier, some of the damaged circuits in the brain. So we use a laboratory rat animal model. This is a rat brain. You're looking at the front and the back. The rat receives a small, what we call mild traumatic brain injury in this area on the top and the side of the cortex. And then we study an area not right where the injury occurred, but behind it. This is that visual processing area you saw in some earlier slides. And what we're able to do is take a little bit of that brain and keep it alive in a dish for about 12 hours. And using a series of micropipettes, glass hollow tubes, we impale individual neurons and analyze their electrical signaling as they talk to each other. We then try to induce changes in the communication strength between the neurons, what's known as synaptic plasticity, which is the basis, we think, of learning and memory in the brain. We do this in a normal control situation, and we do it after the brain has been injured to see if we can modify it. When we look at the normal brain through the microscope with a variety of methods that let us visualize, using fluorescence microscopy, certain molecules that tell us if the brain has been injured, in the mild TBI, we see certain reactions. These are glial cells that have become reactive and other types of cells that tell us indeed we do have the injury. But the brain is not dead. The brain has been injured. There is indeed some necrotic tissue in the area, but the other circuits in the brain are behaving abnormally that can lead to all sorts of problems for the patient, or the rat, as it may be in this case. And so what we do is study a phenomenon illustrated here known as long-term potentiation or long-term depression. That simply means the increase in the strength of synaptic communication over time, or the decrease. I implanted the pictures of the discoverers of this phenomenon. This is Tim Bliss, who originally discovered this, and Terje Lomo. This is Maso Ito, who discovered the flip side, called depression. And this is Serena Dudek and Mark Baer, who studied how this works in the brain during learning in a part of the brain called the hippocampus. All of these people have contributed mightily to our understanding of the mechanisms at the biological level in the brain that lead to learning and the formation of memories. So we study this phenomenon in individual neurons, and we know as the neurons learn, even in a dish, they change their connections within each other, as illustrated through this picture that shows lengthening and shortening of these little processes. These are the processes where the synaptic input comes in. So here's the experiment we do. We take the normal brain or the injured brain, we collect control information, activating the synapses for a while. We then stimulate the brain with a certain pattern. But we don't just change the frequency of stimulation that is typically done in most clinical situations, like from once per second to 10 per second, 100, 200. We change the pattern, the temporal pattern. And the reason we do this is a long story, which I could share with you later, but I basically went to bed for 20 years after doing experiments all night and listening to loudspeakers generating the activity in living brains, and the noisiness of it stuck with me. And I thought there must be something to the noise. So let me show you how we do this. First, this is a regular pattern of activity. I hope you can hear it. Not very loud. Very regular, okay? That would be the one illustrated here. Now I'm going to play what's called a Poisson distribution, a statistically very irregular pattern of activity. Same frequency. Sounds a lot like what you heard earlier, doesn't it? This is what we're playing through the hollow glass tube into the brain to see how it affects this synaptic plasticity. And what we found is there are dramatic effects. These show the responses of many neurons when we have perfectly regular activity, slightly irregular, or very irregular. And this shows the responses where they go up, that's in the red, the long-term potentiation. When they go down, the blue, that's long-term depression. And this is comparing across a whole series of these experiments in normal control animals. And then if we look at animals who have had a brain injury, as illustrated here, same thing, perfectly regular, highly irregular. You can see the amount of plasticity is different. So what we found is the brain does care about pattern, even the normal brain. And every neurosurgeon would accept that, I think. But we had to find it experimentally. And we found the injured brain and the normal brain respond very differently to those patterns. What we hope to do is harness the brain's intrinsic ability through patterns of activity to release chemical neurotransmitters, so you don't need exogenous drugs to replace those that are released all over the brain, but you'll get specific release by the neurons in response to the natural pattern at the area that you need it. And I just want to end up by saying, we're also looking at a variety of other things in the neurons, the amount of calcium inside the cells. We have found, for example, the injured brain, the amount of calcium in the cells is much higher, and that can lead to all sorts of changes. So it's given us some strategies to try to treat the injured brain. And it was mentioned in the introduction that I went to Antarctica recently. It's actually not quite right. My lab went to Antarctica. They were going in the middle of winter, and I was offered to go. I said, you know what? It was the Antarctic winter. Why don't my students and postdocs go instead? And they did. The seas were 40 feet. The winds were over 100 knots. They had all kinds of scopolamine patches on them so they wouldn't throw up. It was quite an adventure, 4 days across, but they got there. We worked for a year preparing for this and sent down all the equipment. They went to Palmer Station right here on this ship, set up the lab, spent 3 months in the dark and cold studying this animal. This is an Antarctic ice fish, a notothenioid. They are at great risk of becoming extinct. They represent 90% of the biomass in the Great Southern Ocean. They live in freezing water. They have antifreeze in their blood. They're the only vertebrate that does not have hemoglobin in their blood. They don't need it because the water's so cold, the oxygen just diffuses out of the blood. The problem is they're dying. You know why they're dying? Because the water's getting warm. You know how warm it is? 0. That's too warm for their brain. And so we did a series of studies where we're recording the activity of the neurons in the brains in these animals while they're swimming and slightly changing the temperatures. And sure enough, what we're seeing is the patterns are changing, and it's the irregular patterns of activity changing from regular to irregular that seem to be modifying their behaviors. So what this will result in, we're not sure yet. We don't think we can therapeutically treat all these animals with deep brain stimulation, but we do think we're getting evidence that will be important to try to inform us about our decisions in this area. And I just wanted to close with this to show, I think, the beauty of science that goes from the whole global health system, the environment, and understanding medical applications, from understanding basic biology and principles that are so informative. Thank you for your attention.